All right, sounds good. For those who don't know me, I'm Zach Wilson. I'm Assistant Biosafety Officer here at University of Colorado Anschutz Medical Campus. And it's my pleasure to introduce Dr. Thomas Campbell, who will be speaking on the status of current COVID-19 vaccine candidates. Tom graduated from the University of Texas Southwestern Medical School, where he also received his MD. He's been a professor of medicine and microbiology at the University of Colorado School of Medicine here at Anschutz since 1995. He's also a past chair of our IBC, so thank you for that service. He's also the Chief Clinical Research Officer and the Associate Dean of Adult Health Research at the University of Colorado Hospital for UC Health. And his practice is in clinical infectious diseases at the University Hospital, and his research focuses on the evaluation of medicines to treat viral diseases. He's the principal investigator for the AIDS Clinical Trials Unit. He's also now the PI for the Moderna COVID-19 vaccine trials that are happening at Anschutz as well. And I've really been looking forward to seeing this talk. It's quite timely, so let's all welcome Dr. Campbell.
So this is a snapshot of what COVID-19 looks like in Colorado as of yesterday. And so as of yesterday, we had over 176,000 confirmed cases. If you do the math, that comes out to a little over 3% of the population has had a confirmed case of COVID-19. Of course, there are probably other cases of unconfirmed SARS-CoV-2 infection that didn't result in COVID-19 illness. So we're probably, upwards of 10% or more of the population infected. And as a result, there have been over 11,000 hospitalized cases. That's roughly a little over 6% of all the confirmed cases. And among the confirmed cases, there have been 2,651 deaths. So that's 1.5% of all the confirmed cases. And these graphs here are from CDPHE website, and they just show you what the trends have been over time. The graph on the top right shows the confirmed cases on a daily basis. You can see that on the right-hand side here in late October through the current date in November, we've had this huge spike in cases that really dwarfs the number of confirmed cases that occurred back in March and April, which on this graph just look like little blips. Of course, there is a lot more access to testing now than there was, and that's certainly part of it. But testing doesn't affect hospitalization rates. You can see the hospitalization rates on the bottom left side, and you can see that the confirmed hospitalized cases is significantly higher than what we had back in March and April. And as you can see on the bottom right, the graph with the red bars, these are the deaths. And testing access doesn't affect death either. And fortunately, deaths are not as high as they were back in March and April. I think there's several factors that contribute to that. Here at our hospital, University of Colorado Hospital, if we just look at the death rate amongst the people who are admitted to our hospital, it's about half now. Now it's about half of what it was in March and April. So I think we've made significant improvements in how we treat COVID-19 that is affecting mortality. Next slide, please. And this just shows what's going on in our hospital system. So this is for all of UC Health. And you can see that across our system, we have now 361 people hospitalized compared to our peak of 263 back in late April. Next slide. So to really get out of this epidemic or pandemic and the impact that it's having, we need to develop immunity in our population to SARS-CoV-2 infection and COVID-19 illness. And so one question is, well, is that going to be possible? What are the prospects for doing that? So SARS-CoV-2 is a recently emerged human coronavirus. Of course, it came from bats into humans just in the past year or so. But there are four endemic human coronaviruses that have been in humans for thousands of years or more. And so humans have a good deal of immune responses already in our population to these viruses. And when they infect us, they cause the common cold. It's very unusual for them to cause a severe respiratory illness, such as can occur with SARS-CoV-2, although it does happen sometimes in immunocompromised hosts. So this slide shows some data on what happens when volunteers are experimentally, intentionally infected or inoculated with a endemic human coronavirus. In this case, it's human coronavirus 229E. And so what they did was to take 15 volunteers and to squirt an inoculum of 229E into their nostrils, and then over time measure antibody responses. And there are the antibody responses over time are pictured in these graphs. There's both the filled circles and the empty circles. So the filled circles are people who had pre-existing antibodies, either IgG or IgG or IgA, as shown here. And the dark circles are people who didn't have detectable antibodies. What you can see is that the people who had antibodies didn't get infected, and their antibodies didn't change much over time. There were 10 out of the 15 people got infected, and 8 out of the 10 who got infected had cold symptoms. But the infected group had lower baseline antibody levels than the uninfected group, as shown on these graphs. And in the infected group, there was a rapid increase in antibody levels, both IgG and IgA, within about 2 weeks. But then that declined over time. And by a year later, the antibodies in the infected group were very similar to the uninfected group that had pre-existing antibodies. Next slide. And then what they did in the same experiment is a year later to re-challenge these people with the same 229E coronavirus. Only 14 out of the 15 were re-challenged, and 11 out of those 14 became infected, but only 1 out of the 11 this time had symptoms of infection. And the duration of shedding of virus in respiratory secretions was shorter. So in those who were infected the first time, shedding lasted on average 5.6 days, and then after the second infection amongst those who were infected, it lasted 2 days. So about less than half of the duration of shedding. This has implications because shorter duration of shedding, one would think that would also decrease the duration of infectivity or transmission potential to others. Next slide, please. So I've mentioned there are these 4 endemic coronaviruses, and I've on this dendritic tree here, I've indicated them with the red boxes. And shown on this tree are the genetic distances between these viruses and each other, as well as the recently emerged coronaviruses. So this includes SARS-CoV-2, as well as SARS-CoV or just SARS-CoV-1. And those two are fairly closely related, as you can see here. And then there's the MERS coronavirus, MERS-CoV. This is the Middle Eastern respiratory syndrome virus. And it's more distantly related to SARS-CoV-2. Next, please. This graphic representation on the bottom here, in a color-coded way, shows the sequence homology for various open reading frames in the coronaviruses. So the open reading frames that are indicated here, 1AB is a large open reading frame for non-structural proteins. So this includes proteins with enzymatic functions such as polymerases and helicases, proteases. S is the spike protein, so that's the major envelope protein that's on the surface of the viral envelope. It's the protein that engages the receptor on the host cell to allow infection. There's another envelope protein called E, a matrix protein M, and then a nucleocapsid protein that helps package the viral RNA genome. And what you can see in this colored chart is the relatedness of these different viral genes across the spectrum of coronaviruses as related to SARS-CoV-2. So SARS-CoV-2 is very closely related to SARS-CoV-1, and so you can see by the dark red bar that goes across the top of that there is sequence homology across all of these proteins with SARS-CoV-1, although the spike protein, the S protein, it's a lighter shade of red. And as we go down from top to bottom across these other coronaviruses, you can see that there is less sequence homology, although there's some sequence homology, for instance, with the 1AB proteins, but the spike protein has a very limited homology across the other coronaviruses. So the bottom line is, from this, we don't expect a lot of cross-protection from previous exposure to the endemic coronaviruses in terms of cross-protection to SARS-CoV-2. And this is further demonstrated on the next slide, which shows in a color-coded way homologous as well as heterologous antibody responses across the coronaviruses. So that the red squares indicate a strong homologous response. So this is when one virus is tested against, or antibodies from the same virus, two different patients with the same virus are tested against each other. So strong homologous responses across the spectrum. But there's limited strong heterologous responses, as shown by the green squares. A lot of places where there's weak responses, and then a lot of white squares where there's no data. You can see there's not a lot of data for SARS-CoV-2, but since we know that SARS-CoV-2 is related to SARS-CoV-1, if you follow the row across the bottom, second from the bottom for SARS-CoV-1, you can see that there's not a lot of cross-protection with the other 4 endemic human coronaviruses, but perhaps there is a little. Weak, in a couple of cases, some strong. So we can't expect that our whatever herd immunity we currently have existing from thousands of years of these 4 endemic coronaviruses circulating amongst us to provide much protection against SARS-CoV-2. So then how do we get to that level of needed immunity to perhaps convert SARS-CoV-2 to the behavior of an endemic human coronavirus? Next slide, please. So the paradigm for this is that you would have an infection. That would be the initial infection. You would develop an antibody response. Over time, that antibody response might wane, and you then might be susceptible to get a second infection. But that second infection, because of the memory cells in your immune system, the illness is less severe, as we saw with the 229E human coronavirus experimental infections. And so next slide. Some politicians, anyway, I don't think any serious scientific people have proposed that we could do this just by letting the pandemic run its course so that we have widespread naturally acquired immunity to SARS-CoV-2. However, that would come at a fairly high cost. I showed you on a previous slide what the current state of the pandemic is in Colorado, and I've repeated that here. And then I've made a couple of calculations and extrapolations. Next slide. We show that if we let this run its course and let's say we wanted to reach 2 million confirmed cases, that would still be less than half the population of Colorado. The population of Colorado is currently about 5.7 million. And it would take, at the current infection rate of roughly 5,000 cases per day, it would take us an additional year to get a total of 2 million confirmed cases. And at the current rates of hospitalization and the current death rate, that would result in 125,000 additional hospitalizations across the state and 29,000 additional deaths. So yes, it's probably possible that we could get widespread naturally acquired immunity and get the needed level of immunity across the population to convert SARS-CoV-2 to an endemic coronavirus, but that would come at a very high cost. This is just what would happen in Colorado. Colorado is like about 2.5% of the entire population of the U.S. So multiply that by 40, and that's what the cost would be across the country. Next slide, please. So the way then that we could achieve this without having such significant morbidity and mortality would be to have a vaccine that could provide induced immunity to SARS-CoV-2. And the paradigm for that would be that you would receive an immunization, you would develop an antibody response to that. As a natural infection, that antibody response could diminish over time, and you could still be susceptible to infection at a later time, but because of memory cells in your immune system, that infection, if it occurred, would be less severe. Next slide. So as a way to get this done, the U.S. government created what they call Operation Warp Speed. So the top colored bar in this figure shows the typical process for getting a vaccine from the laboratory to use in the population. And according to this graphic, that takes about 73 months, so over 6 years, for that to happen. And the various steps in that process are color-coded, and the legend is shown below. You can see that there's a variety of things, including preclinical evaluation, then the phase 1, phase 2, and phase 3 clinical trials. And just to get to the end of phase 3, clinical trials typically takes about 4 to 5 years. And then after that, there's about 15 months are required to ramp up production of the vaccine and 6 months to get it distributed across the population. So what Operation Warp Speed proposed to do was to condense this process. And this was done not by cutting corners in any way, but by doing rather doing all of these steps sequentially, to do some of these steps in parallel. So for instance, rather than completing phase 1 before moving on to phase 2, phase 1 and phase 2, as well as preclinical work, is being done in parallel. And phase 2 is also being done in parallel with phase 3. And then manufacturing, rather than waiting till the end of phase 3 to start manufacturing, they've actually begun manufacturing to a large scale prior to getting phase 3 results. So in the hopes that if the phase 3 results are good, then there will be a large quantity of vaccine to rapidly deploy and begin distribution to the public. And so they estimated then that by doing this, they could condense the usual 73 months to as little as 14 months to completion. And completion here would be, would end once the vaccine has been distributed. Next slide. So that's great. That's a great way to think about conceptually how to get the job done quicker. But as I said, we can't cut corners, and in doing that, there are important questions that we need to consider about any candidate SARS-CoV-2 vaccine. Next, please. So number one, which viral protein to target? Next. And is the vaccine safe? Next. And when we think about safety, we need to think about both short-term and long-term safety. Next. And we need to consider with safety whether there is any antibody-dependent enhancement. So there are some viral infections where there is antibody-dependent enhancement. In terms of human infections, the one where that is most clear is in dengue fever. So dengue is an infection caused by a mosquito-borne RNA virus. It's transmitted in tropical areas of the world, and there's 4 main circulating strains of dengue. And if you get a vaccine for one strain, that vaccine, if it only covers one strain, doesn't protect against infection from other strains, although there is some cross-reactivity. And what happens in that scenario is that the person can still get infected, but the cross-reactive antibodies, although they are not protective, they actually enhance viral infection of cells and lead to more severe dengue hemorrhagic fever. And so this is not observed in human coronaviruses, although there is a feline coronavirus where this does happen. So far, we've not observed it with SARS-CoV-2, and it's not been observed in the animal models of SARS-CoV-2 either. So hopefully that will not be the case, but it's nonetheless something that needs to be evaluated as part of vaccine safety. Next, please. And then it's important to consider safety in special populations, including pregnant women, children, and immunocompromised individuals. Next. So how do we define efficacy. So we're going to be talking about vaccine efficacy, and how does that get defined? Next. One way to define it would be to prevent infection. So prevent confirmed infection of SARS-CoV-2. Next. But another way would be to prevent COVID illness. So who cares if you get SARS-CoV-2 infection? As long as you don't get sick from it, then it's okay. Next. And then once we've figured out how we're going to define efficacy, we need to determine whether, in fact, the vaccine is efficacious. Next. And we need to know what efficacy is in those individuals who are at greatest risk of bad outcomes from SARS-CoV-2 infection. And these groups include the elderly, as well as some racial and ethnic minorities and immunocompromised persons. Next. And then lastly, we need to know whether vaccine efficacy is durable. As we saw from some of the previous slides, natural endemic coronavirus infection immunity tends to wane a bit with time. And so will we see this with vaccine-induced immunity? Next slide, please. So this is a schematic of the coronavirus virion, and this applies to all the coronaviruses. They have the same sort of structure. So in terms of SARS-CoV-2, if we're going to create a vaccine, we would want to use a vaccine that's targeted to, or at least start with using a vaccine that's targeted to those viral proteins that are actually in viral particles. And so this shows some of the proteins I've mentioned before: the envelope protein, the spike protein, the nucleocapsid protein, and the matrix protein. Well, it turns out that from what we know from other coronaviruses, next slide, the spike protein is the one that is most affected by neutralizing antibodies. So in the people who have recovered from a coronavirus infection, the antibodies that prevent the virus from infecting cells in the laboratory in tissue culture are those antibodies that target the spike protein. So the spike protein has been the major target of our vaccine effort so far. Next slide. So talking a little bit more about how we would evaluate a vaccine and how we would assess efficacy. So I've mentioned this a little bit already. We can assess efficacy to prevent infection, to prevent disease, such as preventing hospitalization or severe disease, or even preventing intensive care unit admission. And of course, preventing death would be another measure, and then preventing transmission to others could also be a benefit of a vaccine. So these are all different measures that could be used to determine vaccine efficacy. It doesn't matter which of the measures is used. The way the vaccine efficacy is determined is by calculating the risk in the unvaccinated group minus the risk in the vaccinated group, so the difference between those two divided by, or as a fraction of, the risk in the unvaccinated group. Or another way of thinking about it is that the vaccine efficacy is the proportionate reduction in whatever the outcome is among the vaccinated group. Next slide. And you can see here what vaccine efficacy is for some other common viral diseases that we use vaccines for. You can see that for some viral infections like hepatitis A, hepatitis B, varicella, we have some very effective vaccines, whereas, say, for influenza, our vaccine efficacy is a bit lower than that. Now, it's next slide. it's important to know that vaccine efficacy, as we've been talking about and as we will be talking about, is what's measured in clinical trials. So clinical trials are very well-controlled experiments that aim at obtaining very accurate data about a number of different outcomes, and both for safety and efficacy. Next, please. But the term vaccine effectiveness is when the outcomes are measured on a population level. So in the lay press, you'll often see efficacy and effectiveness used interchangeably or synonymously, but in fact, to the vaccinologist or the public health official, these are two different things. They may measure the same outcome, but one is measured in a tightly controlled clinical trial, whereas the other is measured on a population level after the vaccine has been deployed to the population. Next slide, please. So there are over 198 candidate COVID-19 vaccines worldwide. These are in various stages of development. There are 44 of these that have entered clinical trials in humans. And here in the United States, 6 of them have entered the final stage of clinical trials, or phase 3 clinical trials. So phase 3 is the final stage of clinical trials where the intent is to prove vaccine efficacy and to assess vaccine safety over a large group of people. And the 6 trials that are either ongoing or will soon be ongoing in the U.S. are listed in this table. And you can see the different companies, but you can also see that they have different technologies. So the AstraZeneca and Janssen vaccines use adenovirus vectors to deliver DNA that codes for the coronavirus spike protein. AstraZeneca uses a chimp adenovirus vector, and Janssen uses a human adenovirus vector. Both of these trials are in phase 3, and enrollment is ongoing. They are enrolling here in Colorado. AstraZeneca is enrolling in the Fort Collins area at UC Health North, and Janssen is enrolling at 2 sites here on the Anschutz campus, one through the pediatric infectious disease department, and the other at the Rocky Mountain VA. Novavax and Sanofi are both protein subunit-based vaccines. So these are recombinant spike protein is made in the laboratory and purified, and that's what makes up the vaccine. And that's similar to the way that we make hepatitis B vaccine or the newer version of the shingles vaccine. These phase 3 studies are still pending enrollment. We hope to start Novavax in the first week or two in December here on the Anschutz campus. And I was just on an investigator's call for Novavax before I joined this meeting, and they're estimating perhaps the first week of December we'll be able to start. Sanofi will be a little bit further than that. And then I've highlighted here on the bottom the Moderna and Pfizer vaccines. And I've highlighted them because these are the ones that are furthest along in development here in the U.S. These are both mRNA-based vaccines. So for these vaccines, a synthetic messenger RNA that codes for the coronavirus spike protein is what's injected. Now, the messenger RNA is not the immunogen, but the messenger RNA is taken up by cells and then translated into spike protein, which then serves as the immunogen. And the Moderna trial has completed its phase 3 enrollment. I'll show you more details about that in a minute. and the Pfizer trial has almost completed its phase 3 enrollment. Both of these trials have been conducted at sites in Colorado. The Moderna trial was, we were a site here on the Anschutz campus. Next slide, please. So let me, I'm going to go into a bit more detail about the Moderna vaccine. And first here I'll show you the preclinical data from a non-human primate model. So these are rhesus macaques. And again, the actual product that Moderna is testing, they call it mRNA-1273, and it encodes a stabilized pre-fusion conformation of the spike protein. And what you can see here is that when these monkeys were given the vaccine and then experimentally inoculated with SARS-CoV-2, that you can see that the monkeys who got the control or phosphate buffered saline PBS, they had fairly high levels of viral shedding shortly after inoculation. That then diminished over time, and then in their BAL as well as that was BAL on the left side as well as nasal swabs on the right. So BAL is bronchoalveolar lavage. So that's what's going on in the lung. And the nasal swab, of course, is what's going on in the upper respiratory tract. And they tested 2 doses of the mRNA-1273, 10 micrograms and 100 micrograms. And you can see that both in the BAL fluid as well as in the nasal swabs, there was much less shedding of viral RNA in the monkeys who got the vaccine. And I should emphasize that the y-axis on these graphs is in a log scale, so it's a 10-fold difference for each increment on the y-axis. So these are pretty big effects. Next slide. So in a phase 1 study, the mRNA-1273 was highly reactogenic. So what that means is it gave people symptoms after they got injection. And it's kind of hard to read all the print on this slide, but these are various symptoms including pain, swelling, redness at the injection site, flu-like symptoms, myalgias, muscle aches, headaches, fatigue, fevers. And what you can see at each of these bars then represent the different doses that were evaluated in the phase 1 study. 25 micrograms, 100 micrograms, and 200 micrograms. And the first column is what happened after vaccination 1, and then the second column is what happened after the second dose of the vaccine, vaccination 2. And what you can see is that there were very high rates of these symptoms, particularly after the second vaccination. Next slide, please. And this shows the phase 1 results for immunogenicity in humans. And so particularly what this demonstrates is the induction of neutralizing antibody. So the induction of antibodies that then in the laboratory inhibit virus replication. And you can see on the left-sided graph the results for neutralizing antibodies against a SARS-CoV-2 pseudovirus. And on the right, it's neutralizing antibodies for SARS-CoV-2 whole virus. You can see that on the left-hand graph, the different doses, 25, 100, and 250 micrograms. And then the far bar on that right side of that left-hand graph is what's observed in convalescent plasma. So convalescent plasma is the blood plasma from people who have recovered from a COVID-19 illness. You can see that there's quite a wide range of neutralizing antibody titers in convalescent plasma. but both the 100 microgram and the 250 microgram dose produced titers of neutralizing antibody in the pseudovirus infection that were either in or close to the top quartile of what's observed in convalescent plasma. And then on the right-hand graph, same sort of thing, but this time for whole virus neutralization, just comparing the 25 microgram and 100 microgram doses, and just a few data points for convalescent plasma. Next slide, please. So older people are at greater risk for getting severe COVID-19 illness, and we know from other vaccines, particularly like influenza vaccine, that older people don't respond as well to new immunogens because there is senescence in the immune system. So it's important to know if any of these or how these candidate vaccines work in older individuals. So this slide shows again the pseudovirus neutralization as well as the wild type virus neutralization that occurs after 25 or 100 microgram doses of the mRNA 1273 in people who are 56 years of age or older. And so it's broken down by people in the 56 to 70-year-old range, as well as then people who are older than 71 years. And what you can see on the left-hand slide is that there is again a dose response, so there's more neutralizing antibody with the 100 microgram doses, but you can see that the levels that are produced for the 56 to 70-year-olds and the 71-year-old and older groups are very similar, and they are also very similar to what was produced in the 18 to 55-year-old group, which is on the right-hand side, just to the left of convalescent plasma. And again, they're at or near or in the top quartile for convalescent plasma. And again, on the right-hand side is the whole virus neutralization. And you can see that for the two different age groups, after a 100 microgram dose, the levels of neutralizing antibody are essentially identical to what was seen in the 18 to 55-year-old group. Next slide, please. So to help evaluate the COVID vaccines, a part of Operation Warp Speed was to create a network of trial sites across the U.S. These are funded by the NIH through the NIAID as part of a new network called the COVID Prevention Network. And here at the Anschutz campus, we actually have two COVID Prevention Network sites for conducting these trials. One is based in the Division of Infectious Diseases in the Department of Medicine, and the other is based in the Section of Infectious Diseases in the Department of Pediatrics. Next slide, please. And so the first study that the COVID Prevention Network supported was the Moderna study, again evaluating the mRNA 1273 vaccine. Moderna dubbed this study the COVE study. Next slide. So this is a phase 3 randomized, blinded, placebo-controlled study. Participants were randomized to receive either the vaccine, which was the 100 microgram dose of mRNA 1273. As you saw from the phase 1 data, 100 micrograms produced higher levels of antibody than the 25 microgram dose, but the levels of antibody between 100 and 200 micrograms were similar, so 100 micrograms was what was chosen to go forward. It's Participants are randomized to get vaccine or placebo at an equal ratio, and there's 2 doses, and it's given on day 1, and then 4 weeks later, again on day 29. And the primary goal was to evaluate the efficacy of this vaccine, not to prevent infection, but to prevent COVID-19 disease as compared to placebo. The whole Moderna has made the entire protocol available to the public, and you can go to their website and download the whole protocol if you want details. Next slide. But some of the key entry criteria here are that it enrolled people who are at high risk of SARS-CoV-2 infection because of their occupation or other circumstances. It included both healthy adults or adults with pre-existing medical conditions that are stable. It did not test people for antibodies for SARS-CoV-2, although people with a known history of SARS-CoV-2 were excluded. It did not include individuals who were immunosuppressed, although well-controlled HIV infection was allowed, and there were some restrictions around the timing of other vaccines. Next slide, please. So let's go back to vaccine efficacy. So for this study, vaccine efficacy was the primary endpoint, and this was looking at symptomatic COVID-19 disease. So what that meant is that to qualify as having symptomatic COVID-19 disease or qualifying as a primary endpoint, participants had to have both systemic symptoms, at least one, and at least one respiratory symptom, and have a positive SARS-CoV-2 PCR. The bar going into this was actually set pretty low, and the hypothesis was that we lost the slide. The hypothesis was that the vaccine efficacy would be greater than 30%. So, I showed you vaccine efficacy for other vaccines in the 80 to 90% range. 30%, excluding 30% is setting the bar pretty low compared to those other vaccines. And for determining how many cases that they need, and to get the 90% power to see a difference, they estimated that vaccine efficacy would be at least 60%. And with 60% vaccine efficacy, they needed 151 cases of COVID-19 to be able to detect a difference. And so that's how the study was designed, was to be able to detect 151 cases of COVID-19. And the study had 2 planned interim analyses by a Data and Safety Monitoring Board. The first one was planned to occur after 53 cases of COVID occurred across the 2 arms, and the second after 106 cases across the 2 arms, and then the final analysis would be when 151 cases occurred. Next slide, please. So this shows enrollment at our site here on the Anschutz Medical Campus. And what I want to point out is that we did enroll a fairly diverse population here, as you can see by the pie chart on the right. In almost 49%, a little over 48% of our enrollees were from non-white minority groups. So we enrolled over 27% Hispanics, over 8% African Americans, and over 7% Native Americans. And this was very important because we wanted to ensure that we had a diverse study population. Next slide, please. And this shows enrollment over the entire country. So enrollment began on January 27th, and it was completed on October 23rd. So it took less than 3 months to enroll over 30,000 volunteers at 99 sites across the United States. I mentioned the diversity here at our site in Colorado. There was also good diversity across the entire study population, with 37% of volunteers coming from racial and ethnic minorities, including over 6,000 Hispanics, 3,000 African Americans, and 7,000 people who were 65 years of age or older. And over 95% of individuals have now received 2 doses of either the vaccine or placebo. By the end of next week, that'll be 100%. Next slide. So there was a press release earlier this week, just 4 days ago, and in this press release, from Moderna, they said that the DSMB has completed their first interim review. Remember, I said that it was planned after 51 cases, but in fact they reviewed 95 cases because enrollment happened much faster than was anticipated. I should also mention that the DSMB was convened by the National Institutes of Health, so this is not in any way connected to Moderna. So it's an independent, completely independent DSMB. And what was remarkable was that amongst these first 95 cases, there were only 5 cases in the vaccine arm and 90 cases in the placebo arm. And there's a very significant, very low p-value for the difference between those 2 arms, and the measured vaccine efficacy at this early time point is 94.5%. So highly effective and well in that range of highly effective, highly efficacious vaccines. They also have examined some key secondary endpoints, and one of the key secondary endpoints was severe COVID illness. So severe COVID illness is illness that requires hospitalization. And there were 11 of the cases in the placebo arm were severe versus 0 in the placebo arm. Now, there's no p-value associated with this. The numbers are very small, although they do appear to be quite different. And importantly, there were no deaths in either arm. They've not released much in the way of safety data yet. They've told us that that the grade 3 adverse events that occurred in greater than 2% frequency, including injection site pain, fatigue, myalgias, arthralgias, headache, pain, and erythema, all listed there with their frequencies. And so Moderna, based on these early findings, plans to submit to the FDA for an emergency use authorization by the end of this month. The clinical trial is continuing, and all participants are still blinded, and the plan is to reach the 151 mark of confirmed cases, which is the planned final analysis. Moderna actually anticipates that they'll hit 151 cases before the end of this month. Moderna has said that they can produce 20 million doses of vaccine for distribution in the U.S. before the end of this year, so by the end of December, and up to 1 billion doses by the end of next year. Next slide. So Pfizer, I mentioned, also has a RNA-based vaccine, and these are the immunogenicity data from their phase 1 study. And they actually evaluated 2 different mRNAs in their phase 1 study. I'll just call them B1 and B2, and they encoded slightly different parts and conformations of the spike protein. And what they found was that, shown here with immunogenicity, this again is neutralizing antibody titer, similar to what I showed you for the Moderna vaccine, is that across comparing the 2 different vaccines, B1 and B2, there was no difference. And looking at younger people and older people, there was no difference in neutralizing antibody titers after 2 doses of the vaccine, as indicated by the arrows along the x-axis. Next slide. There were differences, though, between these two vaccines in terms of reactogenicity. And so you can see the B1 vaccine on the top panels and the B2 vaccine on the bottom panels. And this is specifically for the older age group. And you can see that there were more side effects, more adverse reactions with the B1 vaccine compared to the B2 vaccine. So based on this, Pfizer went with the B2 vaccine in their phase 3 study. Next slide, please. And so we also had a press release this week on Tuesday from Pfizer that announced the results of the final endpoint analysis for their trial. Their enrollment began on this exact same day as Moderna, July 27th. They conducted their study at 150 sites, and it was multinational, so it included the U.S. as well as Europe, South Africa, and South America. And as of Tuesday or Monday, they had enrolled over 43,000 out of a planned 44,000 participants, and most had received, the vast majority had received their second vaccine dose. They also had a good racial and ethnic diversity, and they had reached 170 confirmed cases of COVID-19. They were also planning to have 150 cases, roughly similar to Moderna. And of these 170, 162 were in the placebo arm and only 8 in the vaccine arm. So vaccine efficacy, which was measured beginning 7 days after the second dose of vaccine, was 95%, with a very low p-value. And their vaccine efficacy was consistent across the various age, gender, race, and ethnicity strata. And specifically in the older adults, vaccine efficacy was over 94%. And they reported their safety in the same sort of fashion that Moderna did, which was grade 3 AEs that occurred at greater than 2% frequency. And the only thing that hit that mark was fatigue and headache, as shown here. And they submitted to the FDA for an emergency use authorization today, and they plan to produce 50 million doses by the end of December and 1.3 billion by the end of 2021. Next slide, please. Novavax, as I mentioned, is coming. We hope to start this study within the next 2 to 3 weeks. And it is, again, a protein-based vaccine. The design is very similar to what I've shown you for Moderna and Novavax. Next slide, please. I'm sorry, Moderna and Pfizer. Next slide. Could I have the next slide, please? Looks like we're having some technical difficulties with the next slide. So I'll just pull it up on my own. Oh, there it is. Okay. So the next step after these vaccines are approved will be for distribution. And so CDPHE has developed a phased plan for COVID vaccine distribution. And this is available on their website as well as more information. But what this chart shows is that there will be a total of 6 phases. In phase 1, we'll focus on healthcare workers, first responders, and then persons who are in long-term care facilities, so the very highest risk individuals. Phase 2 will include individuals in congregate housing situations as well as essential workers. And phase 2B will include people who are living independently but have high risk issues that put them at increased risk for severe COVID illness, including age, obesity, and medical conditions. And then phase 3 is when it will be available to everybody else in the general public. So we anticipate that phase 1 will start in early January, and phase 3 could perhaps start by the end of the second quarter of 2021. So perhaps in May or June. And it's estimated that it'll take almost the entire year of 2021 to get the vaccine administered to the entire Colorado population. Next slide, please. So in summary, what we've seen is that 2 independent phase 3 clinical trials of candidate COVID vaccines with similar technology, both mRNA-based technology, have found early efficacy in the order of 95%, which is really quite amazing. We've gone from first recognition of this illness in humans to highly efficacious vaccines in less than 12 months. This is early vaccine efficacy, but it's in the range of what we've seen for other highly effective vaccines. We don't know what long-term efficacy and safety will be, and that still needs to be assessed. So these trials are still ongoing. The process for regulatory approval has been initiated, and as I've said, we think that these will be available by early 2021. We've seen there's a coordinated plan for how they're going to be distributed. Other vaccines are still pending evaluation. It's important that those trials continue because we will likely need more than just 1 or 2 vaccines to get the pandemic under control and get high levels of vaccination throughout the country. So I will stop there and I'm happy to take any questions.
Some couple of COVID tend to be more resistant to infection with SARS-CoV-2.
No, that's a very good question, and I have not seen that. Based on what we know about cross immunogenicity, we would expect that there would be some protection. We do know that people with SARS-CoV-1 are resistant to subsequent SARS-CoV-1, but I've not seen anything about cross-resistance to SARS-CoV-2. I would add that there's good evidence of natural immunity with SARS-CoV-2. There have been some documented cases of reinfection, but those have been just a handful, and when you consider that millions of people have been infected worldwide, it seems to be a pretty rare event.
That appears to be the only, did the people in these studies actually go out in public, and how do we know that they were even exposed to COVID-2?
Well, the people in these studies were recruited because they were people who were at risk for exposure to SARS-CoV-2. So specifically, healthcare workers, elementary, high school teachers, people in retail, people in other public-facing companies and transportation, people working in crowded workplaces. So they were recruited because of what they do and where they live and how they live. They were not asked to change anything about their behavior. They were not asked to intentionally expose themselves to SARS-CoV-2. The reason that we know that they were exposed is because their there were 95 people across the study that got infected. So we know that there were at least 95 exposures.
And then do you feel do you expect side effects to differ with mRNA vaccines versus protein-based vaccines?
We'll have to see, but I think that the short-term side effects, the reactogenicity, may turn out to be greater with the RNA-based vaccines. You know, that reactogenicity that I showed you for the Moderna vaccine is quite high, and it may turn out to be less with the protein-based vaccines, just because there's less protein. You know, it's protein that's being injected rather than being expressed. So otherwise, I think that the longer-term or potentially more serious side effects will be the same.
And then last question is, do you feel the vaccine alone will solve the COVID crisis?
Well, the vaccine will only solve anything if people take it. So a vaccine that is not taken up very well and sits on the shelf won't do any good. So if there is high uptake of the vaccine and we get a large portion of the both the U.S. population as well as the world population vaccinated, then it will eliminate the COVID pandemic. Now, it's not going to eliminate COVID-19 as an illness. I think that will stay with us probably for a long, long time. But we will see a lot less of it. And because we will see a lot less of it, we will be able to return to life as we once knew it in February.
That was our last question, sir.
Great. Well, thank you for having me.
Excuse me, Cynthia, there's still 2 more questions.
I'm not seeing them, so.
I'll go ahead and read them. Do you anticipate that individuals will be able or will benefit from receiving more than one form of the vaccine?
We don't know what would happen. I don't think anyone has yet. We know from other vaccines that getting more than one form of the vaccine is not harmful. So, for instance, the older shingles vaccine called Zostavax was a attenuated virus vaccine. The newer one called Shingrix is a subunit vaccine, and it's more effective. So we have revaccinated people who previously got Zostavax, and there's been no consequence to that. So I don't think will be any harm, but it probably won't be necessary as long as the first vaccine was effective. It's possible that there could be more short-term side effects, the reactogenicity effects, the pain, swelling, redness, and flu-like symptoms.
Okay, and then this is the last question. Will all vaccine technologies other than messenger RNA require cold chain delivery?
Well, I think all vaccines in general, not just COVID vaccines, require some type of cold chain. Protein doesn't last forever if it sits at room temperature. So at the very least, they require some degree of cold chain. Between the 2 RNA vaccines, there are differences. So the Pfizer vaccine requires storage at minus 80 Celsius or on dry ice. But the Moderna vaccine is more easier to deal with, and Moderna vaccine can be stored at refrigeration temperatures for up to a month. And so I would expect, protein vaccines would be similar, requiring at the minimum to have refrigeration storage, and the same for the adenovirus vaccines. Those are vectors, so they have to remain not replication, but at least transfection competent, and those would likely have to be stored at least at refrigeration temperatures.
Last minute one came in. How do we market the vaccine so people will trust its safety?
Yeah, so that's a very good question, and I don't think there's a single answer to it. I think there are multiple answers. The first thing is people have to be confident in the process that led to these vaccines. As I've shown you, under Operation Warp Speed, the time for getting things done was quite compressed. But again, I don't think that any corners were cut. So, we need to the public that corners were not cut, that we did evaluate these things fast, but we also evaluated them properly. And a big part of that will come once we actually see all the data. So right now, all that's been made public are these press releases. Eventually, the data will be published, and we'll have more access to it, and we'll be able to dive down deeper into what the safety looks like. The second thing is that we need to know the long-term safety, so we'll need to continue to follow vaccine participants. And then the third thing I'd say is that was part of the importance of enrolling diverse populations in these studies, is that we know that certain groups are at high risk of COVID, and there are groups that are in great need of receiving the vaccine. And so we need to be able to inform people in those groups that they were tested amongst people just like them, and that we do have data both for efficacy and safety in people like them.
Okay, great. Well, Dr. Campbell, thank you for taking the time. We know how busy you are. You are definitely in the thick of things and have been for many months. So thank you for the work that you do. I know as a member of the Anschutz campus in the University of Colorado, I certainly appreciate the work that you do, and I'm happy to make whatever small contributions we can from environmental health and safety. So thank you again, and great talk.
Great. Thanks, Denise. And
thank
Zach,
you, Dr.
Cynthia, thanks for inviting me.
Thank you.